Good day. In the current study, we looked at the phytosanitary status of stone fruit nursery trees and propagation material. The fungal isolates that we identified were then further tested for their pathogenicity status. Plum and nectarine varieties were sampled from 3 nurseries. Upon sectioning of the plants, one could see typically from the base a brown discoloration moving from the crown upwards, showing that open wound that is exposed to the soil, infection can take place. The seedling rootstock variety in the middle shows no discoloration, obviously because the roots protecting the crown area. On the right hand side are typical symptoms that we saw of these nursery trees. Infection in the graft union and also infections taking place from the pruning wound on the rootstock. In total, 1,080 trees were investigated. Of these, 22% harbored canker and wood rot pathogens and 24% harbored cylindrocarpon-like fungi. For the canker and wood rot pathogens, there were no difference in the cultivar combinations. But for the cylindrocarpon-like fungi, we did find significant differences. This graph illustrates the infection level of the different sign and rootstock combinations of cylindrocarpon-like fungi. For rootstock 1, in the plum combination, it had the highest infection levels. It is interesting to note that the nectarine seedling rootstock had the lowest levels of infection, nectarine 1, 2. Results from the propagation material showed that of the green sign buds isolated from, only 0.4% harbored canker pathogens. Isolations from dormant sign buds showed that a higher percentage of buds were infected with canker pathogens, although relatively low, 1.2%. In looking at rootstock material, dormant rootstock shoots were collected from different nurseries and isolations made and showed that 6% of these shoots harbored canker pathogens. Isolations from ungrafted rooted rootstock plants showed that 11% harbored canker and wood rot pathogens and 6% cylindrocarpon-like fungi. The ungrafted rooted rootstock plants had again the exposure to nursery soils. The relevance of the fungal isolates were determined with pathogenicity trials. 37 canker species and 1 wood rot species were wound inoculated on shoots of sunkies and African rose shoots of these trees. After 4 months, the shoots were harvested, split lengthwise, and the lesions were measured and compared to the agar inoculated control. All of these species proved to be pathogenic. The cylindrocarpon-like species, 11 of them, were tested in 2 ways. Firstly, by wound inoculating dormant shoots of 2 rootstock cultivars, Maridon and Mariana. And these rootstock shoots that was inoculated were evaluated in the same way, measuring the lesion lengths. All of the species showed to be pathogenic in comparison with the agar inoculated control. Whole plant assays were also conducted on 2 rootstock cultivars, Atlas and GF677, obtaining tissue culture plantlets from a producer. Unfortunately, these plants or these results could not be used because the measurement of the reduction in root mass was not consistent. And that is most probably because the 3-month period of which these plants were incubated together with a millet seed inoculum in the soil was too short to determine that. What we did observe was some root symptoms with a dark brown discoloration or black spots on the roots of inoculated plants versus the control that had no pathogen. To conclude, within this project we found canker and wood rot pathogens present in healthy nursery stone fruit trees. Propagation material is one source, but also infections can occur via the pruning wounds on the rootstock of the nursery trees. The canker and wood rot species were shown to be pathogenic. Mother block orchards need to be assessed in terms of their phytosanitary status in whether they are continually used as source of propagation material. Nursery trees require effective pruning wound protection on the rootstock wound. And lastly, within this project, we investigated a small sample of tissue culture rootstock plants where we compared their phytosanitary status in relation to normal production plants, and they were indeed cleaner. And this is definitely the way forward in the production of clean rootstock material.